Okay, welcome everybody. We're going to talk about a few different things today. First, we're going to start with some of our NASH open clinical trials, and specifically ones that we haven't talked about before because they're a little bit different. So we'll quickly go over those, and then Andrea is going to talk about two major projects. One is eliminating Hep C across the Mount Sinai healthcare system, and then an automated FIB4 calculation and entry into our EHR. So just really quickly to get our mind thinking about NASH, as you know, that when you follow patients with NASH, what we're learning is that the course is not always linear. And so patients can improve, they can remain stable, and about 40% will go on to develop fibrosis progression, with about 5% developing cirrhosis-related complications. And not all patients with fatty liver disease are the same. It's important to distinguish patients with simple steatosis or bland fat from those with inflammation, and both overall mortality and liver-related mortality is increased in patients with NASH. But the most important thing is that it's really the fibrosis that matters. So even if you have steatohepatitis in the absence of fibrosis, it does not really matter. And so it's key in all of our when we think about these patients, the most important thing to say is, okay, you have fat, but the question is, do you have fibrosis? And there are a number of ways to distinguish that. We can look at non-invasive tests of steatosis, right? We sometimes see it on conventional imaging, ultrasound. Now we have CAP, so we can we have on FibroScan, and then there's MR spectroscopy, which is part of a lot of the clinical trials now. We'll talk a little bit about that, and then multiparametric MRI, which Amon's going to also talk about. And then there are some non-invasive estimations of fibrosis, the NAFLD fibrosis score, and then the FIB4, which is why Andrea is going to talk a little bit about having that auto-calculate in the EHR, so that we are able to identify patients at risk for having significant fibrosis at kind of a more population level. A FibroScan we do in the office all the time, and of course MR elastography. And so the NAFLD fibrosis score, there is a calculator. This is frequently used to identify patients for clinical trials. The NAS score, and if you have a NAS score of 5, that's usually the threshold that will qualify you to be in a clinical trial. So what they're looking for is a NAS score greater than 5 plus some degree of fibrosis. And now in earlier trials, they just kind of said, okay, if you have a NAS score of 5, you're in. And now you have to have activity in all of the different fields. In some of the earlier studies, in fact, GenFit, in the Golden trial, they didn't require ballooning, and so you could have like 3 from steatosis, 2 from inflammation and qualify, but now you must have ballooning on the biopsy for inclusion into all of these clinical trials. So the non-invasive scoring systems, the NAFLD fibrosis score can be calculated, and as I mentioned, the FIB4 score. I think those two are the most commonly used when we talk about NASH. The NAFLD fibrosis score is calculated. You can easily get it online, but the only thing I wanted to point out is the albumin grams per deciliter. When you look in EPIC, it's reported as milligrams per deciliter. You need to move the decimal point over 1 when you do the calculator. Otherwise, you will get an incorrect score. We currently have, these are our current open trials for NASH. NASHNet's really more of a registry, but in terms of therapeutic trials, there are 4 that are currently open. We're going to talk about mostly NGM18 because that's new. I will briefly talk about Madrigal, and if time, we can certainly talk about the other ones. As you notice now, you cannot get into these trials. Most of them are F2, F3. Madrigal luckily allows some patients with 1 stage fibrosis if they have some other parameters like ProC3 elevation that is a propeptide that suggests fibrosis. And so what we've got right now, and I think Shanti and Meredith, oh, they're here. So Shanti is the coordinator for NGM and GenFit. Meredith's not here, right? Okay. Okay. Yeah, it doesn't matter. But point is that we have our coordinators here. And you can see how we're doing right now in these trials in terms of the number of patients screened, those that have been already enrolled in screening, and our screen failure rate. So look at that screen failure rate. That's kind of in line with what you see across the country. About 65–70% screen failures for NASH. We're often surprised when a patient will have a FibroScan value of 9 even and not have enough fibrosis to get into these trials. We actually won't even screen a patient unless their FibroScan is greater than 8. Like, don't even bother. They're not going to get in. And then, of course, there are some issues with the central reader, sometimes different than what our pathology thinks. But that being said, the important point is that there's a high screen failure rate for NASH studies.
How do you define screen failure? Just anyone who's referred, or someone who's gone through non-invasive, or someone who's had biopsy?
They've been biopsied. So these are the screen failure rates where you've been biopsied, sent to central read, and the most common reason for screen failure is not enough fibrosis on liver biopsy. We had one in Madrigal where her GFR was off by, like, her GFR is 49 and she needed 50, but that's rare. Most screen failures are related to biopsy reads.
And for NGM, was really weird when they first started the study. They had very peculiar kind of criteria where they had to have an AST greater than 20, and we had patients who had biopsy proven F2, F3 because their lab didn't say AST greater than 20. They said no, but they recently amended that. So and we've seen wild things where I had a patient who had F4 by all reads on the biopsy on the FibroScan, and we sent her for an F4 study. They said F2. So it can be absolutely maddening as the PI for some of these trials.
Yeah. And so I think we do it aggressive, and our coordinators are great at doing aggressive pre-screening. We won't progress to biopsy unless we see kind of high FibroScan score, elevated numbers, cardiometabolic stuff that is, suggestive of their likelihood of having NASH is very high. So with that said, we're going to talk only about 2 trials because these are newer, and certainly we can discuss why FGF19, but I'll let Amon take over and talk a little bit about this drug.
So thank you all for coming again. We'll talk a little bit about FGF19. This is the NGM compound that's out there. And so this is some of the mechanism of action and the rationale. So initially, the way they discovered this, they were looking at bariatric patients who were losing weight after their bariatric surgery. They saw this signal for FGF19, and that's why they started investigating it and creating analogs. And this is a downstream target for FXR, so like obeticholic acid is an FXR agonist. And it's really about that FGF secretion in the intestine. And it's a metabolic regulator that inhibits gluconeogenesis, promotes glycogen synthesis, and it inhibits insulin-induced hepatic lipogenesis. And also, it works on regulating bile acid synthesis. So it inhibits bile acid synthesis from cholesterol via cytochrome P457A. And one of the markers you look at, so this crazy long thing we call C4, this 7-alpha-hydroxy-4 cholesterol 3-one. That's, if you read the literature, you see that called C4. And that's kind of one of the intermediates when you go from cholesterol to bile acid synthesis. So it's something they often will use to kind of track if it's working.
So you're looking for elevations.
Precisely. So you know you're kind of shunting the other way. So NGM is the company that came up with the compound 282 for the phase 2A studies. This is one that they've engineered, and they claim that. So in the HCC world, we'll talk about FGF19, and it's a bad thing. But they claim that they've deleted the mitogenic region for this. They haven't seen any signals for HCC in any of the animal models or any of the human trials as of yet. And some of the investigators claim that actually will help protect against HCC. I don't know if the data is anywhere close to support that, but it's something that they claim. So they're kind of 2 different studies that Harrison.
All small proteins.
Mm-hmm. It's injectables. Yeah. They're engineered small proteins. It's a daily injection. That's the one downfall. of this medication. So that's something we always talk about our patients, and we've been trying to target our diabetics who are constantly doing injections. So I'm going to kind of combine the two studies that Harrison put out. The initial one is the daily injectables looking at 3 milligram, 6 milligram, and placebo dose. And the primary endpoint was the absolute change from baseline at week 12 in liver fat content on imaging. He also did another 12-week study where they used liver biopsies. That was the hepatology paper. So I'll kind of lumped them all together for you. But this was the biopsy study where they really just focused on 1 and 3 milligrams. And they did MRI PDFF. And this is the one that actually used the liver multi-scan, which is a Perspectum study that I'll kind of go into later. Perspectum, we're doing work with them. And that's also a non-invasive biomarker utilizing imaging. And the really interesting thing about this is this was the first study to do biopsies after 12 weeks looking for a response. People thought it was pretty wild that they were going so soon to try to find a response here. And they used historical placebo biopsy, which is a little funny, but it's something that we've discussed at the Liver Forum a couple years ago about potentially using that. I think it's theoretically fine for early studies, but I'm a big fan of using real placebo biopsies just because of the variability that we see in our trials. And I put up the data here because it's really impressive that with a 3 milligram dose compared to the historical biopsy, they saw improvements in fibrosis, steatosis, hepatocellular ballooning, and lobular inflammation. And it's really dramatic, the steatosis improvement that they saw. And this is the 2A paper from Lancet, just to kind of walk you through what they saw here. So this is the A portion of this is the percent of responders. And their definition of a responder is people who lost 5% or larger reduction in absolute fat, as identified by MRI PDFF. And you can see in placebo it was only 7% versus 70%, almost 80% in our patients receiving the compound. And then it breaks it down for absolute liver fat content for you and relative change in liver fat content. You can see that gigantic delta here. It's very impressive. And it's pretty well tolerated. So the major side effects were site reaction from the injection, some GI complaints, diarrhea, abdominal pain, and nausea. But very few patients had to come off this medication. It did increase LDL, which we see in a ton of our FXR kind of downstream products. And it kind of makes sense here too, because again, we're kind of stopping that pathway of cholesterol going to bile acids. So we're going to have more cholesterol there. We're going to have more LDL. And it's still unclear whether this is going to be significant for atherosclerosis, because not all LDL is really the same. We have small dense versus kind of fluffier LDL, but rarely do we ever fractionate the difference between the two. In the second study, the hepatology paper, they put these patients on rosuvastatin, and they responded great. And as I've given you guys lectures before, I'm the biggest fan of statins in our patients with fatty liver disease and with fibrosis. There's a lot of data out there to show that it can potentially help reverse fibrosis, improve microcirculation and microthrombi there. I'm always trying to get my patients on statins, so it's not a problem for me. Another nice thing we saw with this study is this pretty dramatic drop in ALT. You can see over here in A at both doses at 3 milligrams and 6 milligrams versus placebo. Same thing with AST, and it doesn't stick around too long after treatment. It goes back up about a month after they were taken off the medication. Really no difference in alk phos. And again, C4 was that target I was telling you about for bile acid synthesis to show engagement of its target.
What happened to their weight?
It doesn't really change too much.
Didn't change, the diarrhea wasn't that much.
No.
But the statin use is an interesting point, because even Intercept, like when you report the reduction in fibrosis, if half of them were started on a statin, are you looking at that subgroup because the effect could be because of the statin? So and they didn't, and that was asked at ASLD, and they didn't have an answer. I don't know if Scott, but so that's one point to keep in mind, because they need to look at that subgroup and tease that.
Do a statin control.
So
That's right.
that's actually what we're doing for the phase 2B study. I'm gonna get to that, but to your point, when we were doing that Flint trial, was pretty obvious who was on medication and who wasn't. You could see that LDL rise. We, at VCU, we were putting them all on the atorvastatin.
Right,
down
right. And
there.
so if all
And
your
it's like-
effect is from the statin because you are treating the hypercholesterolemia.
Well,
It's-
except that there's a fair bit of data that's not well controlled, because the statins per se don't affect NASH, but maybe the combination of statins and the drug
Right.
have a synergy that either one of them doesn't achieve.
Does anybody know what happens to triglycerides? I didn't look. The reason I bring this up, there was just a warning for the Intercept trial of beta-cholic acid for pancreatitis. And I thought, gee, I haven't been looking at the triglycerides. LDL goes up, like do triglycerides go up, and that's the risk factor for pancreatitis. Just an FYI to think about.
I'll tell you, there was a case of pancreatitis in the 2A study, but I have to go back and double check about the triglyceride if there was a signal there. But there was one case of pancreatitis.
You don't think so?
I don't think
I don't-
so, but-
It's just an LDL rise. I don't see the triglyceride, but that's right now they're
Have been-
excluding
Yeah,
everyone with a history of pancreatitis, and have very low threshold to discontinue if they have any signs or symptoms associated with suggestive pancreatitis. The triglycerides is something that typically went up. Okay.
yeah, that's what I don't recall,
Yeah.
but then I was trying to reconcile the
Even
pancreatitis
the
with,
triglycerides need to be really high for it to be a causality for.
And they already come in with elevated triglycerides too, so something we have to be mindful of for these patients. And so now we're doing the 2B study. So it's we're actively enrolling. This is for F2, F3. We just did the questionnaire. They are planning on doing this in cirrhosis, so we will have an F4 arm soon, which is great. And as Mina had alluded to, they have to have a score of at least 1 in each of these categories of steatosis, inflammation, and ballooning to have that gestalt of NASH, because that's really what they're looking for, is NASH resolution. And so it's 3 doses, 1, 0.3, and 3 milligrams, 152 patients. And it's double-blinded also to statin initiation. So we check the blood work, then they come and see us, and is it week 2 or week 4 they go home with the bottles? I can't remember. Second week, yeah. So week 2 on drug, they go home with 2 different bottles. One's marked A and one's marked like B, and then they have statins in them, and so we're totally blinded. But the investigator as to are they being started on statin and at what dose to kind of eliminate.
Who decides that they go on statin?
So it's there's a very specific algorithm in the protocol, and it's the central kind of like lab is doing this for us. Yeah. So they're trying to get rid of the potential noise and also the safety, which was kind of curious. I've never seen any other study do this. But
By the way, there was a paper recently, I think, in Journal of Hepatology, looking in more detail, which is something that was coming for the lipid abnormalities in response to metacholic acid, and they do have a more atherogenic profile. The company had hoped that in aggregate it would be more atherogenic, but they also had intermediate size particles that went out, which is associated with risk. So
it's not as clean as they claim.
yeah, I mean, whatever. It's taken. I have to go back and look at it again. It just came out in the last month. Something we've been expecting. So all the more reason to put these patients, because presumably it's the same mechanism with the FGF19.
Exactly. Yeah, FXR pathway. I would imagine is exactly the
Surprising,
same.
because in rodents, it's clearly anti-atherogenic.
Yeah, but rodents are a totally different ballgame when you talk about atherosclerosis. I mean, to get real atherosclerosis, you need an LDL receptor knockout, or in mice, and so it's always harder with mice.
The other thing that to your point about controls, there is an effort, and I think Arun is trying to drive this, of creating, pooling all the control data from all the trials, so that you effectively have a standardized control group that you can apply to any trial, so you don't have to continue to do foreseeable control trials.
Yeah, and that's what they did for the hepatology paper that Harrison put out. And I think it's great for early on in the studies, but I think once you're getting more advanced, like 2B3s, you really need to think about biopsy, just because it's such a messy disease. This is definitely a tough disease to run trials on. So with this 2B study, so it's a 24-week study, so now we're extending it from 12 to 24 weeks. And really we're looking for improvement in liver fibrosis without worsening of NASH, or NASH resolution without worsening of fibrosis. And so they need to get a 0 or 1 inflammation and a 0 in ballooning to really call it NASH resolution. And as we've all discussed before, the definitions of NASH resolution have changed over the course of these studies. Okay, and I think the Golden 505, like Mina mentioned, was kind of the first one where they changed it kind of midstream, where they changed the primary endpoint. Not that at first it was weird, but it was actually to the benefit of our understanding of this disease state. And they're looking at other kind of secondary endpoints, so the liver fat content on MRI, PDFF as well. And I won't go through this too much. But they're also looking at the combination of those who improved liver fibrosis and resolved NASH as secondary endpoints. So that's kind of the main focus. And then I'm going to quickly transition to our Perspectum trial, which should be coming on later this month. And this is just kind of, again, an overall view of kind of the patient journeys that we're seeing, where we have the patient presenting to clinic with risk factors for NASH, either our clinics or primary care, with features of the metabolic syndrome, and then getting some blood biomarkers, elevations in AST, ALT, some concerns when you're doing a FIB4 score, an NAFLD fibrosis score. We put ELF on here too, just because these guys are from England, they love it. Often coming for secondary care, getting the FibroScan, and then when we have the concerns that they're above 8 or they're having these CAP scores are greater than 280, will often lead to liver biopsy. That's our dirty gold standard as of now, and all the late stage clinical trials are requiring this, but we need to investigate these non-invasive, or we need to investigate the non-invasive testing more, and we're doing that in conjunction with these clinical trials to hopefully get away from biopsy. That's ultimately going to be the question is, once we have drugs on the market, if a beta-cholic acid comes on the market next month, it works in about 23 to 25% of patients after 18 months, by the study. How do I determine if this drug is working or not? When do I call it quits? These are the major questions that we have that the insurance companies are going to want to know, because they're the ones who are going to approve the prior authorizations. So we have to develop non-invasive tests that are more reliable.
Well, the FAST T, the FibroScan.
With the AST?
Yeah. Just got published yesterday. I think it was also Harrison, right, in the Lancet yesterday. Yeah. So AST plus FibroScan.
So yeah, he's a big pusher of the AST over 20 as that actually was what sabotaged us for NGM. What was they were so reliant about this AST. It's like, I got a biopsy that shows F3 guy. Like, I don't like, what does the AST really matter? We know you have active disease. So nothing in NASH holds true for everybody. So I guess what we found over time. So this is corrected T1 is what you'll see, MRI corrected T1 for NASH. And this is T1 mapping, really that measures longitudinal relaxation time in these patients. And it's hopefully this biomarker will help us understand the inflammation and fibrosis that's occurring. It looks like it's pretty good, with the initial data, but the initial data was on very few patients. The first paper was 30 patients. The second paper was only 70 patients. And we're using it in conjunction. The Madrigal trial is using corrected T1. We're using it in the NGM trial, so we're going to get more data. But this is all within the guise of clinical trial, so we're trying to do a pivotal trial to understand its true effectiveness in determining the amount of activity and fibrosis in these patients. This is kind of what the pictures look like. So the first one on your left is the corrected T1, or the T1, which makes water and fat bright. Then they have this, water and fat bright is in T2. Fat will be bright in T1. But they have this weird kind of proprietary T2 that they do the subtraction. The whole point of correcting the T1 is you get a lot of noise from iron. And so that's what they've created, this proprietary kind of software to correct for that. And then initially they were calling it the LIFT score, liver inflammation and fibrosis score. They're not allowed to call it that anymore. They got in a little bit of trouble for that. So again, it'll be this kind of spectrum of disease, and they'll just come up with a scoring system that will do for them. And it is a little biased, their papers, because they're an industry company, and they were saying in some of these papers that the transient elastography measures were only effective in 59% of patients, which I don't think it's that low. I think they were kind of doctoring up some of their data, but that's why we're doing a pivotal trial at major institutions to really understand the truth.
Yeah, the CEO, well, you
Yeah.
You know, he's a real bullshitter.
He just wants to talk about money and not talk about science. And I've had some very
Tell us what you
interesting.
really think, though.
Yeah. So the redder it is, the more activity there is in these pictures. You can see that and it shows you the histology scores on the bottom for the steatosis ballooning, but it's really just a spectrum. So green is okay. The more red you get, the more activity there is, the more fibrosis there is. So we need to validate this thing. It'd be great if we can use it. I'll tell you, I talked to Bashir about this extensively. It was in the radiology. He's very skeptical of this, but I say let's just go in with an open mind. Let's try to find something. The data is the data. We'll make sense of that. So it's a single gated perspective, cross-sectional observational study. So these are patients we want who are biopsy naive. These are people you're thinking about doing a biopsy on for NASH, and so they're going to get paid. It's a 12-month study. There's only 4 of our centers that are doing it. We're just going to do imaging. We're going to do the biopsy. And we're going to get a little bit of blood from them and be able to look at their chart. That's all we're asking to do. It's a very simple study.
And how often are you doing, I guess all your patients who you biopsy who didn't qualify for your trials?
So it'd have to be before. So they need to be biopsy naive. And so I'll just show you this is kind of the plan. Is the patient comes in and you're suspecting NASH, and then we would do the multi-scan first. They have to be biopsy naive. We really want the data to be pure and not kind of swayed by any
But you're not
previous
doing
data.
that many biopsies or you're largely doing biopsies in patients who you think may qualify for a trial.
So anybody we think who would qualify for a trial, you send them. We'll do the scan first. We'll do the biopsy. Then they're out of this trial. Then we get them into the clinical trial.
Unless they
it's
don't,
our
unless they
screening.
don't qualify for the trial, then they stay in this one.
Yeah. Well, this is
just
done.
This is just to correlate.
Oh, well, there's no longitudinal.
Yeah.
It's done. This is a very
This
quick,
is really
easy.
just to do
Yeah, just to
correlation.
correlate it.
Yeah.
Okay.
Yeah.
It's over the day of the biopsy.
Exactly.
Right.
Exactly.
Not over, okay.
So they're just going to get this MRI for free, and we are going to do MR elastography at the same time for free for them. So any of your patients that you're thinking about with NASH, you want to know more about it, and they're willing to do a biopsy, just send me an email. We can coordinate this and take care of it. Where we'll get them the imaging, we'll get them the MR elastography, they'll get paid, we'll do the biopsy, and then if it's eligible, we'll get them into the clinical trial. I think this is probably the best slide to show you kind of our timeline there. And the primary endpoint is to evaluate those with suspected NASH and try to see, kind of predict before they get the biopsy who will have those NAFLD activity scores of greater than 4. They're really focusing on fibrosis level 2 or higher. That's where the drugs are focusing on. So they want to help kind of with that. And they're also going to compare this to MRI PDF, with MRI PDFF, and hopefully with enough MR elastography if we have the data. So it's a really quick, easy study, and it's the typical inclusion/exclusion criteria. We talked a long time at the investigator meeting about how the biopsy needs to be done. That we really want it to be a 16 gauge needle. Percutaneously, but if we're going to do a transject, we can do an 18 gauge, but we want it all from the right lobe. The left lobe typically kind of overestimates the amount of scarring. We don't want any EUS biopsies there. We want to kind of make sure we can compare apples to apples. And then, it's just kind of the typical risk factors here for NASH. And the exclusion is if you had a prior biopsy, we don't want it, or people who are going to go on to liver transplantation. Or anybody who's received an investigational drug for 30 days prior to enrollment, and then just excluding other causes of liver disease. And no cirrhotics, no alcohol, and then no contraindications to MRIs. That's I won't go through all this stuff. But if you're thinking about any of these patients, just send us an email. Especially, we'll take care of the biopsies. We'll knock everything out for you. And then, so thank you for your time. And unfortunately, I need to run to Mount Sinai West. So see you guys later.
Go west.
Drop the mic. Okay, so I'm only going to briefly mention one other study that's open right now. It's Madrigal. It's a thyroid hormone receptor beta agonist. So it's basically liver specific. It's thought that this receptor is really more liver specific, and it's essentially treating hypothyroidism of the liver. In earlier studies, they saw reduced markers of NASH in animal studies, and then I'll just go to the recent study that was published in Lancet, We were part of this study, 36-week multi-center randomized double-blind placebo control. The key is that they're allowing fibrosis in stage 1. So this is the only study we have that's open for stage 1. And so if you have somebody you don't think is quite at that 2-3 level, but they have stage 1 fibrosis, as long as they have kind of cardiometabolic risk factors, they can get into this study.
And this is the phase 3.
This is the 2B, yeah. No, the
2B.
phase 3, yeah, phase 3. So the drug is 80 milligrams or matching placebo once a day in this study. Here they looked at just the fat measurements at weeks 12 and 36 and did a second liver biopsy at week 36. And the primary endpoint in this study was the reduction in the MRI PDFF assessed by hepatic fat at 12 weeks. And essentially, they saw a good effect in the reduction of hepatic fat compared with placebo, as well as some other non-invasive markers of fibrosis, and now we're doing the larger study. So, I think these are the two ones that we have that are open right now. We do have Saniquinvirac, it's CCR2, CCR5 antagonist, so that's another study that we have open. And we also have GenFit, which is the PPAR study. We are trying to enroll more in these studies right now because these are our newer studies. And I think that the bottom line is if you have anybody, if you send them to us, we'll pre-screen them and we'll find the right study for them. Obviously, some people don't want sub-Q injections. That might be a deal breaker for some in the NGM study. So with that, I'm going to pass it on to Andrea and team, who are going to talk about our bold initiative to eliminate hepatitis C at Mount Sinai. Let's see. Was it on the desktop?
Sure, I hope so.
Let's see. Yeah, it's right here. Yep.
Okay, great. So, two projects, eliminating across the healthcare system and automating FIB4 in the health record. So I don't know how many of you heard Barbara Murphy's State of the Department talk yesterday, but it was really quite interesting and a real endorsement for the idea that in the arena of population health, there was a great need for medical informatics and better use of information that's in the health record and getting on top of things and being proactive with information that's available but just kind of laying fallow. So we have a pretty multi-dimensional healthcare landscape. So you've got physicians, but you also have government mandates, which are really important. And I think when one is attempting to improve health, it's important to really keep the government in mind because what the laws are make a really big difference. And then just Mount Sinai is all over the place. So we've got a lot of people to deal with.
Actually, the CDC is also coming out with the same recommendation for universal
Good.
Hep C screening, probably be around the same time.
I'll get to that. Just 1 sec, because it's
Okay, sure.
a slight anticipation. So I realize not everybody is passionately interested in hepatitis C. And so I just wanted to put out the idea that if you're interested in this general idea that you can use in medical informatics to automate case finding, and this can be a strategy for entering people into care, then one of the first questions that you'd have to ask if you wanted to do that sort of thing is what would be a great disease to start with? And one of the reasons to start with hepatitis C is because it's so easy to diagnose hepatitis C. There's a definitive test. So we don't want to only do hepatitis C. What we're really trying to do is develop an approach that we can use for digital case finding across all liver diseases. But if you're trying to diagnose fatty liver, that's far more complicated than diagnosing hep C. So it's kind of like, we'll start with the easy stuff, develop methods, and then move forward. And not only is hepatitis C definitive to identify, but it also has definitive treatment. So I think it's a little bit not clear quite what you do when you are finding people who have diseases, but you don't really have definitive treatments for them. And we don't have that problem with hepatitis C because we have great treatments that are effective almost 100% with only 12 weeks of treatment. And another reason to start with Hep C is because Hep C is still really a killer. Lots of people are dying with Hep C, and I think a lot more people than we understand based on the way that we're keeping our statistics. But even with faulty record keeping, it's still the leading indicator of liver cancer in the United States. And because we're working here at Mount Sinai, is we're developing medical informatics tools, algorithms, digital signatures. We have the opportunity to see how well we're doing, how well the algorithms work, because there are patients, and we can look in the medical record. So it's an opportunity to develop algorithms where you actually can connect back to reality of the record. Yeah, but we hopefully will move on to other diseases. And one of the reasons to do sort of a big picture idea, I think one of the most important reasons to analyze data this way, and hopefully eventually to be able to make information available in large national databases, is the group of people who might benefit from this kind of thing the most are people with really rare diseases, where any institution in its million population may only have 1 or 2 cases, but across the country there could be enough patients that you could actually find patterns of natural history and also responses to treatment. So this is the wonderful team who's working on this project. So I think pretty much you know everybody here. These are the various tasks that people are attending to. So the original aims of a grant that I got from Gilead to eliminate Hep C were first to develop software, and Max has really been the lead on this, working with Lilly, and then to deploy additional staff, which are also supported by the grant, to help with engaging patients in care. And we have a particular focus on 2 patient groups, but that's not really important for what I'm talking about today. So one thing that is important to understand about this Eliminate Hep C project that we've been doing is that it really is a marriage of a research project and clinical care. I know that there has, especially Catherine's, been very interested in whether you could do this where it's just research matched up with research, and I think this came up very much in our conversations with Semaphore. It's very difficult when the goal is to engage people in a research trial. It's quite a different matter when the goal is to engage people in clinical care. So on the research side, we have IRB-approved protocols and funding from Gilead, and on the patient navigation side, we have Penny, who has been doing this as part of clinical management for many years. So, and I just wanted to mention, if people are interested in research funding, this particular announcement to support innovative approaches to identify, understand, and develop strategies for overcoming barriers to the adoption, sustainability of evidence-based interventions. So I think people have always thought if we had great cures, people would be cured. If we had great diagnostic tests, people would be diagnosed. But it turns out that's not the case. And finally, NIH has caught on to this, and they've started supporting projects. And this is what the program announcement says. For many years, researchers have assumed that tools and interventions efficacious within clinical and community-based trials would be readily adopted. However, compelling evidence indicates this is not the case. And I would just mention parenthetically, there was a test of leftover blood that was done up in a Bronx emergency department, and they found that 80% of the people who tested positive for hepatitis C already had a positive hepatitis C test in their medical record. So there's all this information that's just laying fallow, and we wanted to harvest this information. So Currently, the project has evolved. It started just with digital case finding that Max was doing with existing medical records, but subsequently we've also linked up to the Department of Health, sending down names and dates of birth, and then they will look through their database to see if they have any matches. And we're also working with Mount Sinai IT to get patients with new Hep CRNA tests. And this is just a statement from Ann Winters about the New York law that allows us to operate under our activities and get this information and contact patients. So this is Max's algorithm. If anybody wants the details, please stop by. Because I really want to get that I want to get some feedback on. This is a study, just an analysis of 11,000 people that we found whose most recent Hep CRNA test was positive. And the main thing I want to point out is the high FIB4 score. So the patients in our system who have positive Hep CRNA tests that are out there are largely people with advanced liver disease, and we really need to get hold of them.
This is our data.
Correct.
I wonder if that's worth a diesel weight breaker. That's kind of news, right? Because they, all the literature now is saying the majority of new cases of FC have no fibrosis.
That must be European data. I've seen really different data coming out of Europe. Yeah, sure. I don't know when the late breaker deadline is, but
Valentine's Day.
okay, 14th.
Yeah.
All right. So we found these 11,000 people, and under an umbrella of activity that Punny has been doing for a long time, and with merging with this Gilead money, we have actually reviewed close to 5,000 charts and reached out to patients. And of those, about 18% actually could benefit from what we're trying to do here. So it really is almost 900 people so far. One of the important problems that we encountered is that we could not contact patients by phone, almost 37%. And then one of the other problems that we have is that in some cases, unfortunately, we found out that patients had died, and we were unable to ascertain that from the algorithm that we were using to identify people who'd already died. So from this, we kind of got some action plans.
Well, the phone is a problem because nobody answers the phone.
Yeah.
Because they're always speaking Chinese on the other end.
Yeah. So we got this surprise offer of help from Andrew, who has a very big, beefy team behind him and can actually provide lots of help. And so this is our mitigation strategies that we're trying to work on. So as Doug mentioned, a lot of people aren't answering the phone. So we want to see if we can contact them with text messages. So what I've done on the slide here, the things that we're kind of under development and looking for feedback about are in red. So we're trying to identify exactly what wording we want for our text messages. And another issue is we need to work with Mount Sinai and see if they can guarantee that we can do this in a way that doesn't inadvertently cause a text message charge to go to the person that we're trying to contact. We've successfully interfaced with the New York City Department of Health, and we're working with Epic and with Andrew to get all new Hep CRNA positive test results back, and have plans to provide information about testing guidelines and notify providers when a positive patient has an appointment. And I'll get into
it. Because Andrew said he had access to the national death records. Did he?
We have not been able to close that loop, but I'm glad you bring it up. And when we get back to him, I'll ask about the national death records.
He said we do pay for it. That's not how it pays for it.
I think what happened is that the death records themselves are in a state of disarray because of underfunding from the government to maintain the database. So I think a lot of deaths aren't in the national death registry at the moment.
Social Security probably has pretty good records for the over 65
Yeah.
group.
So it's very important. Yes, we'll cycle back to that. So this is tentative text language. I'm following up on results from a medical visit you had at Mount Sinai. Please call me so we can ensure you're receiving the care you need, or respond to this message with your name to verify your identity if you prefer communicating. Thank you. So,
Brilliant.
well, that's an interesting
Probably ought to
idea.
do one in Spanish too, actually. Yeah. Good thought, Joe.
And the last part, you're already addressing them by name, but then you're asking them to text back the name to verify identity.
No, you're not.
No, this is the navigator.
That's the
Oh, sorry,
navigator.
this is an app.
She's giving her name.
So I'm not sure everybody knows SMS is a text message.
Oh.
Via text, yeah, right.
And then you're gonna have to have a reply opt-out to say stop. You have to have like the
Yeah.
ability for them to say,
If you don't want us to
Opt out.
contact, don't
Yeah.
contact, yeah.
Yeah.
Just say no or stop, right?
Yeah, type stop if you don't want
Is there any limit on the time that the test was taken and when they're being followed up?
I think those are
If it's
very,
more than a month or two, they won't even know what visit it was referring to.
well, we had planned to make it a very open thing for the time because we have test results over a long
That's why
period
I'm
of
asking,
time.
because if you
So-
have somebody who you
right,
find the data and they had a positive Hep C 5 years ago, they're certainly not gonna have any idea. They're gonna say, I haven't been to Mount Sinai in 4 years, so.
Well, what do you think is the downside
I-
of that? Yeah,
well,
I
if
don't
they're
know.
not followed by here anymore, they could get mad.
No, I don't think that's necessary.
So when we did that
I would
last
say,
year,
you might just
we
say,
looked to see
from a
if they were
medical
still
visit sometime in
attributed
the
to our
past
doctors.
X years.
okay, in the past.
Do you know what I mean?
Yeah,
To
so-
avoid
Yeah, how come
that.
my doctor never told me that I had
I think in
Hep
that scenario, people are gonna say opt out or not respond. That would be
Yeah.
fine.
Yeah.
If I got that message and I didn't wanna.
Deal.
You think it wasn't meant for you?
Right, you're no longer with Mount Sinai, yeah, you just say stop.
Yeah.
If you're not curious what they're talking about.
C. Right, yeah.
Yeah.
Well, how did the DOH contact them for contact tracing?
Whatever information that they have, I think they're allowed to leverage to try to,
So they call, send letters. and
it's
so-
usually just a phone number that gets reported.
Sorry,
on
Catherine, did you know?
smartphones.
Oh, Ben?
No, I just wanted to ask about, what about Gmail?
Communications.
It's not a requirement to register as a patient, so I don't think we.
I don't know how many percent of patients have emails, I don't know, some of them.
A lot of them have my chart.
I think you're required to have a phone number at a minimum when you register.
Yeah,
But
everybody's got a
everybody
phone.
in theory should have a phone.
I've seen, some emails for that, I just don't know. how
Yeah,
many
they
percent.
ask, but not everybody provides.
Right.
What about my chart? I mean, my chart, patients are actually.
You
signing
know,
up for
we
that.
could certainly put something in my chart, but I think it has not been very effective. I don't,
The ones that are not following up generally don't
Are
have
not
my
on
chart.
my
Yeah.
Yeah.
But a lot of these patients are people who've literally been tested with less than a year and still don't have follow-up.
I mean, it's probably
Just, other
pretty
ways to-
inexpensive to do a my chart option.
Like a-
And especially with Andrew offering to help. So I don't think the reason it had not been prioritized is because it just did not seem in the past it had been a very effective thing to do, but
Just trying to think of other,
Yeah.
modalities.
oh yes, I don't have time to present our snail mail data. We have mailed 800 people and
Yeah.
we got like 4 responses. So it's definitely, we've tested snail mail and it's
Only people
definitely
over age 70. and
not
people from
cost-
there. No, it's true, they love the mail still, they wanna have that paper, they're, age 70 or higher, go with mails.
And wait till I turn age 70.
Okay, I think, let's see, did I miss a slide here? So this gets to the point that Doug was raising earlier about testing guidelines. So we do want to communicate with providers advocating hepatitis C screening. And email is one way that we might do it. I don't know if email is the best way. But we would have some message sort of like this. It's a message from Doug. Director, we're working on a quality improvement program. Then we want this to be what is the current recommendation. And in New York City, New York State, it currently is for baby boomers. There are discussions about whether it should be all adults. And so we were kind of holding off from launching our Hep C elimination campaign until the government decides on what the recommendation is going to be.
Is this to all providers, including specialists, or just PCPs?
Doug?
I think we would aim at everybody. Well, because, so there's gonna be we met with the marketing people and you were there.
Yeah.
Yeah, with David and,
Yeah.
right, and Andrew was there
Yeah.
too, right? Right, so they wanna do actually even a DTC, direct to consumer, and when these universal screening recommendations come out from the USPSTF and CDC.
To everybody, not just to our patients.
Right, everybody actually. So like if you have Hep C, this is your home, you know, like we can fix it, sort of thing. And so, you know, we have the capacity to treat them, you know, and I think, so this is gonna be two-pronged effort. Obviously, we're trying to track down those 11,000 or 9,000 patients in the system, you know, number one. But number two, we're actually going out all the way. So we'll actually tell all the doctors in the system that a universal screening obviously will come from David Rich or somebody, or Vicky Bacon probably. And then also we'll have DTC advertising, because lots of times the patients will drive the doctors to do the right thing.
Mm-hmm.
So
Right.
I just want, the only reason I ask is right now, we're already sending for this population, if they haven't had Hep C screening done, on the Friday, it goes to all employed practices, PCPs only though, or who's coming in that week and if they're due for that. We added it in August to our patient 360 along with like colorectal cancer screening or whatever. So this
Has
is
it
not,
increased testing?
we're seeing now if, since that happened, if we're seeing any uptake. Probably all of these things to get awareness will make them listen more, but yeah.
Yeah, and then we also.
But we're not hitting specialists. That's
Right.
why I asked.
And then the other thing is, there was just a paper that estimated 3 million kids are infected in the US with hepatitis C. Did you see that? 3 million.
It used to be 4 million altogether.
Yeah, it was really close,
Opioid.
probably type 10 million, but probably 3 million kids. So we connected Ethan Cowan to start screening kids in the ED as well. There's a new pediatric viral hepatitis clinic, because now the indication goes down to age 3.
I don't think that's right, 3 million kids. That doesn't sound right.
It sounds a little high.
No,
but
that-
later-
I think that's off by at least 1 0. That,
Doesn't matter.
Yeah.
We have because of the opioid epidemic, all these women with childbearing age. Anyway, so we could try. We'll try to get that going as well. They said they're seeing more Hep C.
definitely,
That's why
if
they're
kids have a parent that's Hep C positive, I think that would be a great way to focus childhood screening.
Yeah,
Because
and
most
so-
of the, especially the kids who are little, mostly they got it from their family.
Oh yeah, for sure.
Yeah.
And you know, Tatiana is obviously working with
Yeah.
Rhoda in the high risk clinics, you know, for the moms with Hep C and treating those that she can. But the thing is, we don't have pediatric follow up on those.
Yeah.
On those kids. Actually. So I don't know. So we're working on that.
So besides the screening, we also want to have a communication about linkage to care. And so it's provides you with a list of your patients who have an Hep C positive test result. And then this kind of gets into some of the other questions, like only an epic, what if the data came to us from the Department of Health, and over what time frame does this need to be a positive test? And then we're asking them, please discuss Hep C treatment with them and refer any patients you would like us to treat to us. So, Mina, I see a look on your face that doesn't
I
look
think when
fully.
we did the chart, a lot had been treated, right? I mean, when we did the whole LabCorp dump and looked at everybody who, for the past 2 years of data had Hep C
No,
RNA.
well, that gets into the time frame because
Right.
we're not we're not going to go with,
This was 2 years past, so say
Yeah,
2016 to 2018.
Yeah.
So many, even if we didn't have another negative RNA on record, the vast majority had been treated. So there's a lot of manual labor to look in the chart to do that, because you wouldn't want to call a patient and say, hey, you have this abnormal, and then they were treated, so you don't even know like the full story. That is just where it gets tricky in the retrospective. The retrospective is tricky. You don't know the context in which the test was drawn. You know, you don't know anything, and so it's a little bit more private than like saying you need a mammogram. So I think, you know, it's the retrospective that I think you got to review the chart before you do blind outreach, because you don't know what was going on.
Well, yeah, the navigators do that, actually.
Yeah, and that we found so many were treated. That's my point. There's a lot of, you're going to get a lot of hits. There's a lot of manual effort to find the ones that were not treated. I'm not saying it's not right to do that, but it's a lot of effort to narrow down. Whereas prospectively, it's much easier. You immediately check it. They have the test. Everybody knows it. You immediately link them to care.
Yeah, those are easy because you get the report.
The ROI is going to be much higher on your prospective.
Oh, yeah.
well, our ROI was not that bad on our retrospective. I did have the data. So we still found almost 20%
Yeah.
that had not been treated. So that's, to me, that's a lot. This is a lot higher than going out and trying to find pediatric Hep C. I mean,
Not pediatric Hep C.
Yeah.
Oh, yeah, that's
Screening.
much higher.
um,
And at $8,000 a pop for Mount Sinai, for 340B money, that's starting to talk real money.
so
So the idea is, even if they're going to orthopedics at West,
exactly.
and their last FC RNA was positive, then we would be in touch with the doctor and say, listen, if you don't want to treat this, you know, your orthopods, please send them to us, you know, so we can fix this.
And
Unless it's
complaints
like-
will be going to Doug, not
And
me.
this is
So
where
we're
your
good.
adoption thing is going to be
Yeah.
the issue, like getting people to do it with all the physician
Well, you
burnouts.
know, you've got to throw a lot of mud up against the wall and a little bit sticks.
Yeah,
So, but hopefully it'll all
certainly won't
coordinate.
hurt.
We'll all coordinate with the PR, marketing,
Yeah.
direct-to-consumer, CDC, USPSTF.
Yeah.
So this, because there's one other topic I want to quickly get to. So system-wide elimination campaign that Doug's heading up is going to get announced. We're trying to figure out what the guidelines, recommendations are going to be. We will be contacting physicians about screening, about treatment. And one part of this that we're going to have to develop with Max and also Andrew, they very much want a medical informatics infrastructure to determine the costs and the health benefits. So we're going to try to build that out. And switching topics, we want to hardwire Epic to calculate FIB4 scores. So we want to have a fully automatic system where a physician does not need to make an order. We will write software to go back for 2 years, and if there's data that is available and allows a FIB4 score to be calculated, it will be calculated and entered into the health record. If it's above 3.25, it will get a red flag, and we're working out exactly what the language is going to be. And similarly, Amon has for a long time been working on a FIB4 EPIC order. So this is all testing that is not specifically ordered, and we would want to hardware it to go forward as well. So it's not just using data that is
It should be like
already.
GFR, it should pop up automatically. Like, with those labs.
Yes, even
Actually,
if
FIB4 order is working. You can order FIB4s now. But FIB4 apparently, because of the way the system's set up, has to go to LabCorp to calculate it and then come back. It takes like a week. It's ridiculous.
What if you get your bloods done through the clinical lab here?
They still have to send it to LabCorp. to
And are you
calculate.
guys finding that like non-liver providers are selecting?
Well, nobody knows about it or not liver providers, but so that's why we're trying to make it automatic. You know, and that's one of the things that we spoke to Andrew about this morning, actually, because apparently he met with Ken Davis and Ken told them to do whatever the liver guys wanted. So, he said sending the
Get the champagne, Doug.
yeah, really. Sending the FIB4 out the lab core is the most ridiculous IT thing you've ever heard of. And then having it come back, it's like, because our lab says it's like copyrighted or trademarked.
Hire a high school student and they can sit there and calculate.
Now, Max can do it automatically.
Yeah, it's
Max,
crazy.
ridiculous.
It's
just a
totally
calculation.
ridiculous. It is remarkable. So for the FIB4s that are ordered by the physician, the cut point is going to be lower. We have a flag in red.
And you have to have just the interpretation.
Mm-hmm. Yes, and
They don't know what-
we're working out exactly what the
It's like
language
GFR. It
should
should
be.
be a list of the numbers and the explanation. Like panic level.
So, yeah, I think according to this big study that was done on NASH patients, the Stellar trial, I think if you have a FIB4 of 2.67, the specificity for having F3 or F4 fibrosis is 93%.
For
So,
the 3.25.
no, for 2.67,
Is for F3 and F4.
F3, F4, according
Why
to,
not use 2.67 in your automated?
I talked to Mean about it.
You said F2, that's why I was surprised. Now you're saying F3, F4. When we talked about it, F2 or like might be too early to trigger like an alarm. You know what I mean?
Yeah.
Well, I think what I said was 2.67 increases the risk of liver-related death by 43-fold. And that's why we use it for a lot of situations where we have a cut point. But this is the biggest study that I have seen, the largest number of biopsies that were analyzed
Negative.
and then linked to FIB4 scores. And that's what they say, specificity 93% for a FIB4 over 2.67 for F3, F4 versus F0 to F2.
So if F3 is your cutoff, see, I was saying, because there's a lot of people trying to do disease detection, right?
Yes.
So you don't want to sound the alarm for like F1, F2, like immediate,
No,
whatever. But
F3,
F3,
F4,
F3, F4, I think F3, if that's F3, F4, then that should be the cutoff.
Yeah, but the question was for the retrospective. I thought you were saying for this, we really had to make it so this patient needs some action. Yeah. That because we're going to be putting stuff into the medical record that the physician may not even know is there. We thought, wow, we really need a high bar
Yeah,
of sense that this is somewhat urgent situation.
This is a misdiagnosis that needs to be dealt with.
Are you going to talk about this as well? Some kind of blanket communication?
You know, I think it's not
No.
we've got the Hep C elimination launch. I think it could be part of that because let's face it, what is the liver disease that we're going to find when we do this? Most of these patients aren't going to be NASH patients. They're going to be people with alcohol-related liver disease, so Jean, prepare yourself, and patients with hepatitis C. So I think, Doug, if you're on board with it, I think that we could put this in with our Hep C launch.
Yeah, we can get it done at the same time. Now that
Well, and to talk about it in the same communication.
Yeah. 2 bullets, make it simple. So otherwise, nobody will. Sure. Okay, great.
Thank you.
Thank you.
Yeah, for retrospective, I think that's right, because if you sound the alarm for people with stage 2 fibrosis, you're gonna get a lot of people who don't really,
I
don't know.
agree with you.
Yeah,
No,
okay, I got confused with your cutoffs when you said that restage.
I agree.
Yes.
But I'm interested in that point. Probably do a lot more with it.
You are gonna have that order now, but you mean the retrospective? That's retrospective. That's the bad. Yeah, moving forward, you're gonna have the 2.6.
Are you concerned about too many false positives?
With what? Well, that's what, with the 93%.
Yeah, but that's in a very specialized group. If you're going to do an umbrella, for example, like anti-mitochondrial antibodies, especially if it's 100%.
Yeah.
But if you screen it
You
in
screen
a,
every, not even just every liver
that's true. The
injury
pretest
patient.
probability.
That's why I just,
Over
this is NHANES data. It's NHANES data that says 2.67 increases liver-related death by 43-fold. I think a 43-fold increased risk of liver-related death is a reasonable standard
I think
for.
it's a reasonable reason to see a hepatologist and get
Yeah.
a fiber scan. You know what I mean? Like to identify early disease. If it's F3, F4, if that's a cutoff, if that's what it is, F3, F4, then I think that's.
No, I think you got to start somewhere. I'm just
Yeah.
saying that there's, you might find a
There might be.
surprising
Well,
number.
we'll be able to look,
like-
right? When you do, well, this would be good stuff. Because
Yes,
we'll be able
exactly.
to see
Yeah, because I mean, I'm
in
just
a
thinking.
total population screen with no enrichment.
But that's why I'm saying, for NHANES data, not selected, NHANES, it increased liver-related, they did prospective liver-related death 43-fold.
But I don't know if the numbers that you're going to get in our lab is going to be the same as NHANES. Because I just think about the patients that I see
Yeah,
that would also be flagged as F3, F4.
Yeah.
A lot of them are like patients on chemotherapy, or patients who've had heart attacks, or patients who, who have
because
hematologic
you're saying
malignancies.
they have low platelets because they're on chemotherapy.
Altered platelets, and they're getting liver tests checked. I don't know if a lot of PCPs are doing as many liver tests as like hematologists, for example. They are always testing them, because of their risk of,
That's a very good
hepatotoxicity.
point. You might get
So you might get these weird pockets
Yeah.
of like cardiology
hemolysis,
and-
people who are hemolysis.
and in room.
You
and
almost
who
don't
are-
want inpatient labs because inpatient
Exactly.
labs, they got a blood transfusion and they got an AST, it's almost like it has to be ambulatory labs.
It'd be much better
outpatient
if it were
labs.
closer to the end. If you get rid of these weird pockets who are likely to order these,
How do you get rid? So we'd have to.
Well, I
so
think
you
we can
subtract
limit,
out.
I think we can limit to
outpatient.
ambulatory labs.
Yeah.
And
that already fixes a lot right there.
That will fix a lot because
but-
the inpatient stuff's going to be like DIC. You're going to get all this crazy
Yeah.
stuff.
And it's possible that we could do an exclusion based on pharmaceuticals.
Okay.
You could do exclusion of an ICD-10.
Maybe diagnosis.
You want to do a exclude by?
I mean,
I
you
hate
can't get
to do
too
that.
too
No,
credit.
I
Yeah.
think I've told you. We looked at 99 charts who had an ICD-9 for Hep A, zero had Hep A. The ICD-9 codes are so bad. There, I think we would be excluding people.
Yeah.
Maybe,
We're talking about
We're
malignancies
trying to,
though, so these
septic
are
shock. I don't know.
right. I think well, if you do outpatient, that helps. And then in the outpatient setting, I guess you could do by meds. You could do by meds.
But just even outpatient would be a big help because I just
Yeah, I
imagine,
think outpatient.
No,
every
it's a great
ICU
point.
patient
I
every
think
single day,
it's a great
something's
point.
going to get
Yeah.
flagged,
Yeah.
You're going to be
Yeah.
inundated
Yeah.
with junk, basically,
Yeah.
Yeah.
So you don't want that, right?
Right.
Because I'm obviously very interested in this because, I think screening is where it's at for fibrosis. Because in alcohol, you can diagnose it very easily, so, and NASH. So I think this is great. But I
We
just,
can wrap our heads around what would be high value things to
To screen
exclude.
out,
Yeah,
yeah.
to screen out. We just should, think a little bit like things that we know cause hemolysis. I mean, like, things that would automatically,
yeah.
Does hemolysis
It's-
raise ALT or
AST?
AST? AST.
and then you also the platelets,
Yeah,
the low platelets
yeah.
is going to be a problem in some of these things. So let's wrap our head around that, just the exclusions.
That'd be helpful, yeah.
Okay, great, thanks.